Operation mechanism. Mother cell production technology. Stem cell manufacturing technology involves the production and manipulation of stem cells for use in medical and research applications. The process of stem cell manufacturing involves several steps, including isolation, expansion, and differentiation. Isolation is the first step in stem cell manufacturing and involves the extraction of stem cells from a donor or patient through procedures such as bone marrow aspiration or umbilical cord blood collection. Isolation can also involve the use of induced pluripotent stem cells, IPSCs, that are generated by reprogramming somatic cells, such as skin cells, to a pluripotent state. Expansion is the second step in stem cell manufacturing and involves the cultivation and growth of stem cells in vitro, using specialized culture media and conditions. Expansion can also involve the use of bioreactors that provide a controlled and scalable environment for the growth of stem cells, and that can generate large quantities of cells for medical and research applications. Differentiation is the final step in stem cell manufacturing and involves the induction of stem cells into specific cell types, such as neurons, cardiomyocytes, or hepatocytes, through the use of specialized culture conditions and growth factors. Differentiation can also involve the use of gene editing techniques, such as CRISPR/Cas9, that can modify the genetic profile of stem cells, and that can enhance their therapeutic potential. The operation of stem cell manufacturing technology requires expertise in biology, engineering, and regulatory affairs, as well as specialized equipment and facilities. The process can also have ethical and regulatory implications, such as the need to ensure the safety and efficacy of stem cell therapies, and the need to comply with international and national regulations and guidelines. Therefore, it is important to develop sustainable and responsible practices in the stem cell manufacturing industry that promote the ethical and safe use of stem cells and that foster innovation and collaboration across different sectors and stakeholders. Stem cell manufacturing technology has a wide range of applications in various fields, due to the unique properties of stem cells, such as their ability to differentiate into various cell types, and their potential for regeneration and repair. Stem cell manufacturing technology is used for the production of stem cells for medical and research applications, such as cell therapy, tissue engineering, and drug discovery. In cell therapy, stem cell manufacturing technology is used to produce stem cells for the treatment of various diseases and conditions, such as cancer, autoimmune disorders, and cardiovascular diseases. Stem cells can be used to regenerate damaged tissues and organs, to modulate the immune system, and to deliver therapeutic molecules to target tissues. In tissue engineering, stem cell manufacturing technology is used to produce three-dimensional structures, or scaffolds, that can support the growth and differentiation of stem cells. To generate functional tissues and organs. Tissue engineering can be used for the repair and replacement of damaged tissues and organs, such as skin, bone, and cartilage. In drug discovery, stem cell manufacturing technology is used to produce disease-specific cell models that can be used to screen and test new drugs and therapies, and to study the mechanisms of disease progression and treatment. Stem cell models can provide a more accurate and predictive platform for drug discovery, and can reduce the reliance on animal models and human clinical trials. However, the use of stem cell manufacturing technology also raises concerns about its ethical and regulatory implications, such as the need to ensure the safety and efficacy of stem cell therapies, and the need to comply with international and national regulations and guidelines. The use of stem cells also raises ethical concerns, such as the use of embryonic stem cells, and the potential for commercialization and exploitation of stem cells.